Good morning, everyone. My name is Trinity Bivalacqua. I'm a urologist at Penn, and today I've been asked to talk about the most impactful basic science papers in the last 2 years. I promise you I am going to do my absolute best to make this as clinically relevant for this audience, but also highlight the amazing science that's been done in both of these manuscripts. These are my disclosures. None of these are related to the topic at hand. So first, the way I'm gonna set this up is I'm actually only gonna talk about two papers. And I chose these two papers because of the impact that they have as it relates to the scientific experimental design, the methods, and the conclusions ultimately that were made. And I also chose them because of the journals they were published in. So this is a paper from Chang, which is looking at the role of mechanical regulated YAP and TAZ in erectile dysfunction models. Now, importantly, this is a paper published in Nature Communications, which is a very high impact journal. I'm going to walk you through all of the figures and give you, after these, give you the clinical relevance, right? So the first figure looked at the role of YAP-JAZ in mechanical stretch smooth muscle cells and how this impacted transcriptional regulation of these targets. This was done via a global transcriptome analysis, and then ultimately bioinformatics was used to look at the penis of a mouse in the erect and flaccid state. So they were able to isolate smooth muscle cells from the penis where you saw it was erect or flaccid. What they found here is that you had a differential regulation of multiple genes with erection. So just the act of blood flow differentially expressed a multiple different transcriptional regulators. They highlight Hippo here, and Hippo is actually one of the upstream regulators of YAP and JAZ. And you can also see that with erection, you have an increase in the upstream regulators of YAP and JAZ. This is actually amazing that they were able to do this. This is single-cell RNA-seq analysis of, once again, erect and flaccid penis. These are very high-impact sequencing projects that were done. And you can see in this plot here that, sorry, I apologize. You can see very clearly the difference in the cell population. So I think the endothelial cells that were isolated in the erection were greater than that in the flaccid state, which makes sense, right? Because you have blood flow activating endothelial cells, which is releasing multiple neurotransmitters, in particular nitric oxide, which you can see very clearly is increased with erection. I'll highlight once again there, they showed that the Hippo pathway was increased with this single-cell RNA-seq bioinformatic analysis. Now, they then said, if we see an increase in YAP and JAZ in our mouse model, what about in different pathologic states, also in the human condition? So this is a tissue microarray where you actually take the corpora cavernosa of human corpora at the time of penile prosthesis. And they did a tissue microarray and looked at TAZ expression in control. These are patients that have no ED, and then patients that have ED, so with and without. And you can see a down regulation in TAZ. They concluded that this may be one of the causes of ED in these men. But they then looked, very importantly, at different knockout models. So one of the things that's so elegant about this paper as well as the other paper that I'm going to present is that they use multiple genetically engineered mouse models to ask the question if is YAP and JAZ, TAZ, involved in penile erection. What they did was they knocked out TAZ, they knocked out YAP. You see, after nerve injury, a reduction in ICP, and if you knock out both, you get no erection whatsoever, suggesting that these are absolutely essential for erection in these models. And then they used pharmacologically, used Verdo to transcriptionally downregulate YAP and JAZ, which showed the same thing. Essentially, making the point here is that it's essential for erections. Now, this is where it becomes more relevant clinically. So once again, I told you I was going to do my best to make this as clinically relevant as possible. They then went on to say one of the most common causes of erectile dysfunction, or the hardest to treat is actually in patients that have had prostate cancer treatment. So one of the things that's novel about this paper is that they looked at multiple models of prostate cancer treatments. First, the nerve injury model, then they looked at radiation-induced, and then they looked at hormonal deprivation or androgen deprivation therapy. And here you can see in the top panel, this is nerve injury model, this is radiation-induced. And what they show very clearly is that with nerve injury at 5 days, 14 days, 28 days, you get a reduction, but then you have an increase in erections. This has been shown previously. But what's important here is in the immunostaining, which is red, you see a reduction in TAS, which then gets restored over time endogenously when you get regeneration of that nerve. Once again, suggesting that innervation is important for this. With radiation, you see the exact same response. Here, once again, this is just a graphically delineating the transcriptional regulators of YAP and JAZ are downregulated and then increase over time. Then they looked at a model of androgen deprivation therapy showing the exact same thing. With castration, you see a reduction in TAS, and then you also see a reduction in expression or erections in those that are castrated. This in and of itself is not novel, but the effect that this has, hormonal manipulation on TAS expression was reduced. And then what they did was they did another series of experiments where they did nerve injury and then showed that there was a reduction in response to stimulation. And then if they downregulated YAP and JAZ with a pharmacological agent to reduce transcriptional activity, they saw no restoration of erection, highlighting the importance of these transcriptional factors in regulating smooth muscle function in the penis. So their conclusion here is that this is important for regulating normal erectile function, but is also involved in the pathological state. Now, this is an interesting part because Wayne Hellstrom's sitting in the room. People in this room probably may remember this. Adrenomegaline, I don't know if anybody remembers, but adrenomegaline was isolated from pheochromocytoma in the mid-90s, and Dr. Wayne Hellstrom in his lab in 1998 showed for the first time that injecting into the penis adrenomegaline causes an erection in a cat model, right? He's looking at me and going, I have no idea what you're talking about, probably doesn't remember, but I remember because I was there doing those experiments. What they showed is that YAP and JAZ actually transcriptionally regulates adrenomegaline. So adrenomegaline, what we thought back then was just a peptide that may be involved for potentially pharmacological manipulation of erection, now looks to be regulating, it's now downstream of YAP and JAZ. And if you knock out YAP and JAZ, you reduce adrenomegaline, and if you restore it, you're able to promote adrenomegaline expression. Once again, they show the same series of experiments highlighting the importance of this peptide. Now, this is where it gets interesting. This is my last slide for this talk. They then asked the question, we all know that in penile rehabilitation, you can use vacuum erection devices to stretch the penis, which is thought to potentially restore a smooth muscle function or protect smooth muscle function after a radical prostatectomy. They then isolated smooth muscle cells and actually used high mechanics and low mechanics stretch in the culture to look at transcriptional regulation of adrenomegaline plus the use of PDE5 inhibitors. I'll make a long story short. What they showed is that when you have high stretch with PDE5 inhibitor treatment in the cells, in these smooth muscle cells, you actually see more expression of YAP and JAZ with activation of adrenomegaline, highlighting that maybe some of the reasons why potentially these pharmacological and devices are improving potentially restoration of function in the penis may be related to this transcriptional factors. So this is their conclusion. I actually didn't even show you half of what they published in this paper, so if you're interested in it, please read it. I'm just showing you the highlights. They actually make the point that erections, when you have smooth muscle relaxation, they're thinking that the stretch and a blood flow shear is activating YAP and JAZ, which is propensitating smooth muscle relaxation of the penis. So I think this is a wonderful article and highlights where our field is moving and the elegant experiments that are done to show this. I'll now talk about one last paper, which is published in Science. Bud Burnett, who's sitting right over there, was the last person that published a paper in Science in our field in 1992. Does anybody know what that is? That was showing that penile erection was mediated by nitric oxide. So we now have a second paper published in Science looking at the role of fibroblasts in initiating penile erection. So this was done from Karolinska group, and they showed that this is a mouse penis, the fibroblast, once again, also using genetically engineered mouse models to tag, fluorescently tag different markers to be able to show where the fibroblasts are. And what they showed is that fibroblasts are predominantly in the penis, mostly around the tunica albuginea, so red is fibroblasts. Green is your smooth muscle and cells. And they showed that with relaxation or blood flow, you see all the smooth muscle cells here, right? But really, what we didn't recognize until now is that the majority of cells in the penis are fibroblasts. So the question is, why are they there? So they used another very elegant experiment where they're able to use light activation of a Creed mouse, and I'm not going to go into the details of this today, but what they showed was with light, when they cree a mouse and tag it with tamoxifen to activate those fibroblasts, they see a reduction in blood flow in the penis. Very novel. No one's even discussed the fibroblasts in regulating blood flow in the penis. They then looked at the effects of this on blood flow again, and showed that it could, that that was my fault. The previous was tagging smooth muscle cells, sorry. Now here they're tagging fibroblasts, and instead of seeing relaxation, they see contraction, right? So why is it contracting? Well, it's contracting because the norepinephrine transporter is actually found on the fibroblasts in the penis. So the next experiment was, what if we inhibit norepinephrine relief from in the penis pharmacologically? And when they do that, they prevent that contraction. So suggesting that some of the sympathetic innervation and activation of sympathetic adrenergic receptors in the penis is actually on the fibroblasts, and that's where it's coming from. Their next series of experiments looked at dynamic changes in cell-to-cell content. Once again, here's the penis in the erect state. Here you can see all the smooth muscle cells, and you see that in the erect state, there's less fibroblasts that are actually present around those smooth muscle cells. Remember, the fibroblasts are there to release norepinephrine, so we don't want them there. They next did another experiment where they used pharmacologically excitatory and inhibitory chemogenetics in the paraventricular nucleus to activate penile erection. And what they showed is that erections were actually increased when you use the pharmacological activation of the paraventricular nucleus, and that when they did that, they saw this increase in they saw the fibroblast activation in the penis. For a matter of time, they showed that fibroblasts are actually increased in aging. So why would it be increased in aging? Because remember, pathologically, you have a reduction. So if you increase fibroblasts, you could be increasing sympathetic tone. So their final conclusion is that with relaxation, you have this less norepinephrine, the fibroblasts are not present, but when you have when this is the flaccid state, excuse me, when you have an erect penis, you see scavenging of those norepinephrine fibroblasts, and actually it could be regulating penile blood flow. So what both of these studies have shown using genetically engineered mouse models, sequencing, is that we can now identify new targets that we need to be thinking about as clinicians to pharmacologically manipulate. I'll end there. Thank you.